Professor in the Arts, Humanities, and Social Sciences. This is a significant occasion for our college, as well as for UMBC, because it recognizes several people and several things. First, it recognizes the outstanding faculty in our college, and I'll say in a few minutes the names of all of the faculty who, over the last decade, have been Lipitz professors in the Arts, Humanities, and Social Sciences. This is also an opportunity for us to recognize the generosity of Roger C. Lipitz and the Lipitz Family Foundation. Nearly 20 years ago, the Lipitz family provided an endowment to recognize and support innovative teaching and research in the arts, humanities, and social sciences at UMBC through alternating 1- or 2-year professorship awards. And for a decade now, we have recognized one faculty member each year as the Lipitz Professor. Previous recipients of this honor have included, and several of the Lipitz professors are here, and I hope that you'll at least raise your hand when I mention your names, have included Professors Carlo Di Clemente from Psychology, John Sturgeon from Visual Arts, Tom Field from Modern Languages, Linguistics, and Intercultural Communication, Jim Grubb from History, Leslie Morgan from Sociology and Anthropology, Raphael Falco from English, Linda Dussman from Music, and Devin Haggerty from Political Science. And before I introduce this year's Lipitz Professor, and each year the Lipitz Professor who is concluding her or his year gives a talk at this time of year to tell you a bit about the research that she or he has been working on. Before I introduce this year's speaker, I want to note that tonight's talk and reception are sponsored by the College of Arts, Humanities, and Social Sciences, the Drescher Center for the Humanities, and the Social Sciences Forum. I will also note that after Professor Waldstein's talk and the question and answer period, I'll introduce the Lipitz Professor in the Arts, Humanities, and Social Sciences for the coming year. The Lipitz Professor for 2015-2016 is Professor Shari Waldstein of the Department of Psychology. A clinical psychologist, Dr. Waldstein joined UMBC in 1993. She is presently, and I will read her multiple titles here to give you a sense of how interdisciplinary and how cross-campus her work is. She is presently Professor of Psychology and Gerontology and Director of the Behavioral Medicine Program here at UMBC, Professor of Medicine at the University of Maryland School of Medicine, and Research Scientist for the Geriatric Research Education and Clinical Center at the Baltimore Veterans Affairs Medical Center. Her research program examines psychosocial factors in cardiovascular disease and brain health and related disparities associated with socioeconomic status and race. Her work in these areas has been funded for over 2 decades by the National Institutes of Health and other sources, and she has a record of more than 100 peer-reviewed publications and book chapters. Dr. Waldstein's collaborative research spans multiple institutions and is highly interdisciplinary, including colleagues in areas such as internal medicine, geriatric medicine, radiology, nuclear medicine, nephrology, neurology, cardiology, physiology, biostatistics, engineering, sociology, and others. Her work also incorporates multiple trainees across undergraduate, graduate, and postdoctoral and junior faculty levels, and I think it's well worth noting that at UMBC, to date, 22 graduate students have completed their PhDs in her lab, with 7 more in progress. Dr. Waldstein has received awards for her research from the American Psychological Association, the American Psychosomatic Society, the American Heart Association, the Academy of Behavioral Medicine Research, and the Maryland Psychological Association. She's a fellow of the Society of Behavioral Medicine and the Society of Health Psychology, and an elected member of the Academy of Behavioral Medicine Research. Dr. Waldstein has served in leadership capacities for multiple professional organizations, including as president of the American Psychosomatic Society, as an associate editor for Health Psychology, one of the premier journals in her field, and as a regular member of the Biobehavioral Mechanisms of Emotions, Stress, and Health Study section for the NIH. Dr. Waldstein's work, in sum, lies at the intersection of the natural sciences, the medical sciences, and the social sciences, and is thus an amazing and wonderful example of the interdisciplinary collaborative endeavors that our college and UMBC prize so highly. Today, as you can see from the slide, Dr. Waldstein will be speaking about cardiovascular risk factors, socioeconomic stress, and brain health. Please join me in welcoming Professor Shari Waldstein.
Hi. Thank you all for coming to this toasty place at late afternoon, late in the semester, and I know we're all kind of aching for that extra time, so I really appreciate that you've given some to me. I'd like to begin with a few thank yous. First as well to Mr. Roger Lipitz and the Lipitz Family Foundation for establishing this wonderful award. Second, to Dean Casper and the Lipitz Selection Committee for choosing me, thank you. And to our department chair, Dr. Chris Murphy, for nominating me. Check's in the mail. Not really. So I've been asked to stand up here by the podium because I'm attached to all kinds of things, and I think so I'm being discouraged from walking around, flailing around, but I do have a pointer and we'll try to point things out. Uh-oh. Thanks. Okay, so I'm going to be talking to you about several interrelated topics today. I'm going to start with a general overview with how we think about brain health. We'll then talk to you about some of the very common cardiovascular and metabolic risk factors that we know are determinants of early brain health. And then I will talk about disparities associated with socioeconomic status and self-identified race with respect to brain health outcomes. When we all think about health outcomes, we're probably much more likely to think about our peripheral organ systems, right? Our hearts. Whoop. Sorry. I'll get used to this. That's a heart. Our kidneys, lungs, livers, etc. How often do you think about brain health? Well, what I will tell you is that every dimension of health related to our peripheral organ systems and physiological systems in turn communicate with the brain and impact our brain health in both a positive or a negative manner. You're all probably very aware of some of the clinical indicators of brain health, which includes stroke and various forms of dementia, such as Alzheimer's disease, vascular dementia, other neurological diseases. We know that these clinical brain health outcomes can have devastating consequences for individuals. So my fellow collaborators and I are very interested in learning about predictors of earlier subclinical indicators of brain health that can start to help us understand what factors are impinging upon our brain structure and function, and therefore our ability to adequately process information much earlier in our lifespans, so that we can think of ways to prevent and intervene so that people do not progress to stroke and dementia. Backwards. Two of the main indicators we use for subclinical brain health are dimensions of our brain structure and function as assessed by magnetic resonance imaging, and also our information processing abilities and how we perform on tests of cognitive function. So the first thing I'm going to do is show you a handful of slides to explain various indices of brain health that we tend to look at. Now, these data come from a long-time study we did down at the Baltimore VA Medical Center and University of Maryland School of Medicine, in which we recruited about 200 squeaky clean, I will call them, healthy older adults, okay? Middle-aged to older adults, ages 55 to 85. I have to qualify that middle-aged thing as I am approaching the kind of cut score for this older adult category. So these are individuals who have been screened for anything and everything you could possibly imagine. Any kind of neurological disease, any kind of systemic medical disease, any kind of major medical intervention that would negatively impact peripheral health and brain. They live in the community. They've driven themselves to our facility for 6 or 7 visits. And we have done a lot of biomedical assessment, cognitive assessment, and brain imaging. So the first indicator of brain health that I'll talk about is white matter disease. Now, the white matter fiber tracts are the myelinated neuronal fibers in our brains that allow different brain regions to communicate to one another. And the gray matter of our brain are our clusters of cell bodies and dendrites. And there are various ways that we can assess the health of the gray matter and the white matter of the brain. You'll see here in patient N. Patient N is a very healthy older adult who has normal blood pressure. You'll see all around here a very clean signal, no evidence of white matter disease. Whereas in contrast, our hypertensive participant has a lot of white matter disease. You can see it here where there's not a clean transition from the periventricular region, from the lateral ventricles here to the periventricular region. You can see a lot of haze here back around the occipital horns, and you see high intensity signal in an ovoid distribution up here in the left frontal lobe white matter. This is a fairly characteristic pattern associated with hypertension. This is another person in our study who is walking around with what we call a silent infarction. This is literally a hole where brain matter used to be, and it's known as a small silent infarction rather than a large clinical stroke. There are ways that you can rate the degree of white matter disease and silent infarction. In this particular study, our neuroradiologists provided ratings. In other studies, you can do, more commonly these days, computerized volumetric assessment, literally of the lesion volumes. So this is just a depiction here of computer identification of areas of white matter disease. You can also go a step further, which we do in some studies I'll talk about later, in which you can literally kind of pull out and examine the integrity of specific white matter fiber tracts to give you an even more subtle indicator of the health of the cerebral white matter. And you can see changes in white matter integrity even in a perfectly normal, healthy-looking brain without clinical evidence of white matter disease. Another index of brain health is how much gray matter you have. Unfortunately, for all of us, over time, that tends to shrink. A lot of you sitting in the audience are peaking as I speak. It's only going to go downhill. Sorry. And here you can see this person has nice, robust gray matter. The lateral ventricles are pretty small. Here they look a lot bigger because the gray matter has shrunk, therefore allowing the lateral ventricles to expand. And here you see some cortical brain atrophy. So this is a view of the outer surface of the top of the brain. And here you see a lot of nice gray matter, some definition of the sulci and gyri. But here there has been atrophic change, which then allows the cerebrospinal fluid to fill in around the perimeter of the brain, along the midline, into the sulci. Okay, so this is an example of cortical atrophy. In our healthy older adults, completely asymptomatic, 77% shows at least some degree of white matter disease. 59% shows some degree of silent brain infarction. Our record was, I believe, 33 small silent brain infarcts in one individual. And almost everyone is showing signs of brain atrophy, okay, on all of these on MRI. So why are these MRI endpoints important? Why do you need to be concerned about them? Very simply, they are pretty strongly prognostic of poor clinical brain health outcomes. So if you're showing these early signs on a brain MRI, you are at increased risk of having the stroke, of developing dementia. Another way to assess our brain health is adequacy of our cognitive function or information processing abilities. And you can use standard interactive and paper and pencil tests to examine different types of cognitive functioning. And examples here of some of the common domains of cognitive function include our ability to attend and concentrate, the speed and efficiency of our information processing, how quickly we can respond, our ability to learn and remember information in verbal and nonverbal modalities, our verbal or language skills, our perceptual and spatial skills, and then one of our personal favorites, executive functions, which are higher order integrative self-regulatory functions that are supported predominantly by the frontal lobes of the brain. Example that's often been given here is kind of the CPU of your computer or the conductor of an orchestra. The frontal lobes are involved in helping us plan, organize, initiate, sustain, and stop our behavior in productive and socially appropriate manner. All right, I messed something up. Okay, there we go. Okay, why do we care about cognitive function? A lot of reasons. Okay, so the adequacy of our cognitive function is an important part of our quality of life, our ability to function day to day, and our well-being. Now, what we argue as a research group is that even very subtle changes in our cognitive functioning can disrupt our daily lives. You don't need to reach the point of impairment. Okay, so think of those of us sitting in this room, for example. We're probably all, kind of above average, right? We're all, students, professors, are doing a great job. Of course, it would be terrible if we were impaired. What if something happened that just rendered you average? How would that affect what you do on a day-to-day basis? Okay, so long before people are impaired, before people are well on the road to a dementia or a stroke, these subtle changes can be happening for a variety of reasons, which I'll talk about shortly. Another thing to consider, as I mentioned before, most of us have peaked and are on our way down. It's well known that there is aging associated with our brains and cognitions, but it's actually still not exactly known what is attributable to primary biological aging processes versus secondary aging, which is the accumulation of risk factors and diseases that we get over the lifespan, which then negatively impact our brains and cognitive function. So insofar as we can identify those factors and control them, we may have much better trajectories of cognitive and brain health across our lifespans. We also know that our cognitive functioning plays an important role in medical decision making, plays an important role in our ability to adhere to treatments, and plays an important role in our health behaviors. A lot of growing, very interesting literature, particularly on executive functions, the self-regulatory behaviors and health behaviors. I believe Dr. DiClemente and his group are doing some work in that realm. Okay, so this is a bit of the what. What is it that's impinging upon our brain structure and function and our neurocognitive function? This is an overly simplistic, overly linear depiction of some of the things that we think are impinging upon brain and neurocognition at the level of the individual. Okay? This does not include any of the innumerable environmental factors that are also impinging upon us. There are many aspects of our genetic profile that will impact our neurobiological propensities that will in turn influence our behavior and lifestyle, how we feel, how we interact, our support networks, how we respond to stress emotionally, physiologically. All of these in turn affect our biomedical risk factor profile and other newer biomarkers like inflammatory processes in the body, changes in hemostasis, blood clotting, and endothelial function, which in turn are going to ultimately lead to the gradual development of subclinical vascular disease, frank cardiovascular disease, and all of these things are impinging upon our brain anatomy and physiology, and thereby our neurocognitive function. And to make this even uglier, since I'm admittedly ugly model lady, these are all communicating with one another. So in our lab, we like to focus on a variety of cardiovascular risk factors. This slide is swiped from Dr. Wenzel in the back. Recognize that? Yeah. We look at a spectrum of cardiovascular, metabolic risk factors, health behaviors that are known to either negatively or positively impact brain structure and function, thereby neurocognition. These cross areas such as variation in blood pressure or frank hypertension. poor lipid profiles, smoking, poor diet quality, obesity, and aerobic fitness. Now the idea is that early on, we are developing these risk factors that will start to negatively impact our cognitive function and our brain structure and function. But this is the beginning of what we call the slow slide down the slippery slope, as people then begin to develop subclinical vascular disease with greater impact on brain and cognition. And then as we start to develop forms of clinical vascular disease, such as heart attack, arrhythmias, you are going to start to approach clinically significant impacts on brain and cognition. And here's some data just to illustrate that in a study that we did with older adults with various forms of cardiovascular risk and disease or absence thereof. These are healthy people with normal blood pressure. Lower scores are better on this test of executive function. So they're doing the best. If you have hypertension, starting to get a little bit worse. These are folks with peripheral arterial disease, which is a pretty advanced form of cardiovascular disease, highly correlated with disease in the coronary and carotid arteries as well. And these are patients who have actually had an anterior stroke. And this is a continuum we see across multiple types of cognitive tests. And in fact, on some of our tests, our Ph.D. folks are really approaching the performance of that of our stroke patients. So I'm going to talk to you a little bit today about some of the risk factor and brain and cognition associations we've seen across several different studies that we've done over the years, including during this Lipitz year. And those include the Healthy Older Adults study I mentioned that was conducted with my collaborators down at the Baltimore VA and the medical school. A lot of work that we've done with collaborators at the National Institute on Aging's intramural research program with the Baltimore Longitudinal Study of Aging, and work that we're involved in quite intensively right now in a socioeconomically diverse sample of African American and white adults in Baltimore City who are part of the Healthy Aging in Neighborhoods of Diversity Across a Lifespan study. I'll talk a little bit first about BLSA. It's one of the oldest epidemiologic studies in the country, started in 1958, and people come to the Hopkins Bayview campus every 2 or 3 years to get reassessed. We've looked at a whole host of cardiovascular risk factors in relation to change in cognition over time in the BLSA cohort. Depending upon what variables we're working with, we might have anywhere from about 500 to 2,100 individuals. You can see there's a very large age span, about half men and half women, but mostly white. I don't have it in this slide, but this is a fairly highly educated group, average probably about 15, 16 years of education. But these folks have had up to 18 years of follow-up across up to 12 visits where they have come in and done biomedical assessments and neurocognitive assessments. Also swiped from Dr. Wendell, thank you. What this slide shows you is that people with greater stiffening of the arteries, measured non-invasively, show more precipitous cognitive decline over time. So the people depicted in the red line have the worst arterial stiffening, higher scores are bad, you see error, and then as they get older, they're getting worse and worse. Similarly, those with greater atherosclerosis in their carotid arteries are having more precipitous cognitive decline as they age. And I could put up maybe 4 or 5 other slides that would mirror this almost precisely. Instead, I'm just going to show you a summary table showing that across multiple dimensions of verbal and nonverbal learning and memory, a multitude of cardiovascular risk factors are associated with cognitive decline. That includes, blood pressure. Pulse pressure and pulse wave velocity are indirect indices of arterial stiffening. Intimal medial thickening is an indirect index of atherosclerosis in the carotid artery. VO2 max is a measure of cardiorespiratory fitness you get when you run people on the treadmill and put the oxygen mask on them. Cholesterol and creatinine, which you can use to compute an index of adequacy of renal function. We've looked at some of these risk factors in relation to cognitive function and also brain MRI outcomes in our VA study, which is also a highly educated, mostly white cohort. They're happy campers, they're not depressed. These are folks who are ages 55 to 85 and slightly overweight, pretty much free of any comorbid disease other than mild hypertension. This is complicated. I'm just going to summarize it for you. What this tells you is that across across many different types of tests of cognitive function, learning, memory, attention concentration, speed of processing, executive function, spatial abilities, so many different risk factors are negatively impacting our outcomes. This includes blood pressure. We do some really neat blood pressure reactivity testing. What we do is bring people into the lab and piss them off and see what happens to their blood pressure. Oh, it goes up. And this predicts poor cognitive and brain outcomes over and above resting blood pressure. And if you think about it, I mean, your blood pressure is varying every time your heart beats, and you know what happens to you when you're under stress. And you'd probably be surprised if you were hooked up to a blood pressure monitor and saw how high your pressure might go. I will tell you that our peak systolic blood pressure response to our laboratory stressor was 100 millimeters of mercury. Yeah, we almost had to discontinue. Effects are worse if blood pressure is poorly controlled, if you have high blood pressure and also central obesity, high or low total cholesterol, higher fasting glucose, even if you're not diabetic, and VO2 max. Relates to all of our brain outcomes as well. So here a radiologist, a neuroradiologist gave us ratings and we computed reliability of degree of white matter disease, silent brain infarction, and our indices of brain atrophy. And you can see across blood pressure reactivity, glucose, and cardiorespiratory fitness, all pretty potent predictors of poor brain health outcomes. Okay, so these are some of the pieces of ugly model that we've looked at. However, the caveats being, again, that I have not even started yet to talk about the environments people live in or sociodemographic context. Are things the same if you're looking at men and women? Are things the same with people of higher and lower socioeconomic status? Whites versus people of color? And the first studies that I talked about were largely done in white, well-educated, and fairly well-off people who volunteered for research studies. So in the next set of work that I'll be talking about, we're going to start to consider whether these various risk factors impact brain and cognition differently as a function of race and socioeconomic status. Socioeconomic status, very complex construct, I'm sure you're all aware. It has been indexed in so many different ways, but some of the most common include how well educated you are, your income adjusted for your household size, your occupation and its relative prestige, family wealth, goods ownership, home ownership, and also what neighborhood you live in. Race we consider a social construct as well, as per many common sociological views in this area, and includes a multitude of variables that impinge upon individuals, that not the least of which are potent stressors in the United States, certainly in Baltimore, which is where we're doing this investigation. Race and SES are some of the most common correlates of health disparities, differences in health outcomes attributable to one's group membership. Some folks refer to these as health inequities, which reflect the idea that these differences are unjust and that everyone should enjoy the health of the most privileged members of our society. Here I'm just going to talk briefly about life expectancy as a function of race and gender. And you will see in the United States, white women are faring the best by far with respect to life expectancy, whereas black men are faring the worst. What's going on in Baltimore? In Baltimore, we are seeing very pronounced socioeconomic differences in terms of life expectancy. What you see here is that people living in Roland Park can expect a lifespan of about 83 years. This is a full 20 years less for individuals living in the Hollands Market area of Baltimore City. And the folks living in Hollands Market are faring comparably to individuals living in Bangladesh or Nepal. If you take a look at what's going on in the Hollands Market area as far as what the area looks like, and contrast that to Roland Park, it may give you some ideas about why this may be. So there are many, many ways in which health disparities may occur at multi-levels, all of which we ultimately believe are translating into biological processes that influence health outcomes most proximally. These influences really span from micro to macro level factors that impinge upon individuals across their lifespan. So you can think about people's genetic profiles, molecular, cellular, organ level function, individual level psychological, behavioral factors, families, your other social networks, communities where you work, your nation, your state, and higher. All these things are going to impinge upon health outcomes and may in part explain why people of color and people of lower socioeconomic status have poor health outcomes. So there is an epidemiological study ongoing in Baltimore City conducted by the National Institute on Aging's Intramural Research Program. The PIs are Dr. Michelle Evans and Dr. Alan Zonderman, and I have been very, very honored to be an extramural co-investigator for this study for many years and work closely with Drs. Evans and Zonderman on various dimensions of these data. And in part, we are trying to determine how various health-related factors explain neurocognitive outcomes in different racial, socioeconomic, gender, and age groups. Let me tell you a little bit about the design of the Handel study. It's a randomly selected cohort from Baltimore City that was explicitly designed to recruit self-identified whites and African Americans from both high and low socioeconomic strata, with about an equal number of men and women in each cell, and the baseline age range began at 30 and ended up at around 64. This is a 20-year longitudinal study that is currently in its third full wave of data collection. It's confusing because it's called wave 4, but I could tell you about that another time. So there are 13 predetermined areas of Baltimore City from which participants are randomly selected. The households are randomly selected, and then individuals within those households are randomly selected. And the baseline cohort yielded about 3,700 folks. And of course, there's been attrition over time, but still in the realm of perhaps, I think about 2,200 in the second full wave, and the third full wave is ongoing. Instead of asking people to come into a medical center, come over to Hopkins Bayview, come over to Maryland, the investigators worked hard to create these mobile medical research vehicles that drive into people's communities. They get a permit set up in a parking lot or some other area, and residents of the community come and hop on the MRV for their testing. So it's a great way to recruit participants who may not choose to or may be less able to make the journey to an academic medical center for this type of assessment. The study spans multiple domains, again, from micro to macro level factors that I mentioned before. Yeah, I'm going backwards. So how do all these risk factors that I've been talking about relate to brain health on MRI and cognitive test performance as a function of race and socioeconomic status? What's the answer? Cop out. We're learning that this is an incredibly complicated question and have been spending a lot of our year learning about the complexities of the question and the answers. I'll be giving you some examples of findings from the study so far. And again, the sample is going to differ a little bit depending on what analysis I'm talking about, but these are baseline data, excuse me. So again, they're 30 to 64 years of age, a little bit more than half are women, a little bit more than half are African American, 39% are living in poverty, average education is about a high school education. This is a measure of depressive symptomatology. The suggested cutoff for suspected depression is 16, as you'll see, pretty pronounced depressive symptomatology. A lot of them are current or ever smokers. And the average body mass index is right on the line of obesity. It's a pretty unhealthy group of individuals, but again, I want to emphasize this crosses all socioeconomic strata. Okay. We've been using structural equation modeling, a statistical technique, to try to inform us on how different types of risk factors may explain the relationship of socioeconomic status to cognitive functioning. And we've been doing this modeling within subgroups of African American men and women and white men and women. What we're finding so far is that it's not easy to get answers, but at least in women, we are seeing that socioeconomic status, as defined by whether or not you live in poverty, your level of education, and your level of literacy, negatively, the lower it is, the more negative an impact on memory and executive functioning. The higher the levels of depression, which then in turn also influence memory and executive functioning. And in men, behavior, lifestyle, health habits are also relevant to determining executive functioning and vice versa. And when I refer to behavior, I'm talking about smoking, alcohol consumption, use of illicit drugs like marijuana, cocaine, and heroin. We tried to fit the cardiovascular risk factors in these models. They were not predictors, much to our surprise. This was a big shock to us because all the cardiovascular risk factors that I've been talking about tend to aggregate in persons of lower socioeconomic status and are also greater in people of color. One thing that has proven quite interesting, these are data with our collaborator, Dr. Regina Sims Wright at Delaware. And what this shows is that in people living in poverty, poor diet quality is associated with worse performance on a test of cognitive function. Higher scores reflect worse performance here. This is another ugly slide. What I'm just going to point out very briefly is that people living in poverty who are African American are most prone to the negative impact of diabetes on cognitive function. And then lo and behold, here in work with Dr. Wendell, we find that folks with greater degree of carotid atherosclerosis are more prone to poor cognitive performance if they are white and of higher SES. Okay, so the story is quite complicated. So we drew more ugly models. This is actually a depiction of the model we're seeking to test in our current grant from the National Institute on Aging that's linked to the larger Handel's study. It's known as Handel's Scan, nicknamed affectionately by one of our participants as Scandals. Yeah. And we are doing a neuroimaging study with a subset of people from the Handel's investigation. So we do recruit on the MRVs, and people come and see us at the University of Maryland School of Medicine to get the brain scanned. And what we will be doing as we accrue more participants is trying to thoroughly test some of these complicated pathways whereby race, SES, and the intersection between these impact behavior, psychological factors, social factors, how they're reflected in the environments people live in, and how this impacts health status, brain structure and function, and therefore cognitive and physical functioning. It takes a village, so I just want to acknowledge my Handlescan collaborators. This is a 5-institution grant, highly multidisciplinary and wonderful, wonderful people. So folks get their brains scanned, and we found out that people in Baltimore have green and blue brains. Can you imagine? Basically, our colleagues at Penn are doing our volumetric analyses, and they fit anatomical atlases to the brains and are able to derive quantitatively total brain volumes, total gray matter, total white matter. They parse out multiple regions of interest. In fact, there are 92 different regions of interest across cortical and subcortical regions. And they also can assess quantitatively the amount of white matter lesion volume in people's brains. So what are we learning so far? You can see here that if you're not living in poverty status, if you're not living in poverty, you have greater brain volume than if you're living in poverty. And these are just box plot distributions, but you can see the whole distribution is shifted down here for individuals living in poverty. Another thing that we have found with my wonderful collaborator, Tule Adeli, here on campus. I've been working together a lot this year on resting state fMRI data, and people living in poverty have lesser activation of their frontal lobe systems. We actually have a lot of other very exciting findings, but they're hot off the press, and I'm afraid if I told you about them, I'd have to kill you. I don't have enough Horcruxes to put you in. We also find that there's an intersection between self-identified race and socioeconomic status with respect to these brain outcomes. If you live in Baltimore and you are white, you have substantially greater brain volume than if you are in higher socioeconomic circumstances than if you are in lower socioeconomic circumstances. Whereas if you are African American, you're doing about the same. And look, those in poverty are actually doing a teeny bit better. Furthermore, if you are African American and you're living in poverty, you have greater lesion volume than all the other groups. Why? This is what we're working on. A lot of different ideas, a lot of preliminary analyses. We think some of our lesion volume findings are likely due to differences in blood pressure and body mass index. My colleague, Dr. Danielle Beatty-Moody, has some very interesting findings showing that perceived racial discrimination is associated with greater lesion volume. And we think this is in part what might be going on with our higher SES African Americans who are finding themselves in workplaces and neighborhoods where they may be disproportionately exposed to discrimination as a function of their race. We're going to be looking across all levels. A lot of these analyses are ongoing, so another time, but again, they cross the traditional and newer cardiovascular risk factors that I mentioned, across our lifestyles, across many different types of psychosocial factors, and also into various aspects of the neighborhoods, which in Baltimore differ tremendously in terms of income inequality, racial segregation, access to health care, access to healthy food and green space. It is complicated. I'll prove it to you briefly. Yeah, this is just for woe factor. Basically here, we're seeing that if you are younger and you have lower blood pressure, which isn't good either, you have greater lesion volume. But if you're older and have higher blood pressure, you have greater lesion volume. So the story is really complicated. We don't have enough participants to tease apart all the different things that are going on in different subgroups, but ultimately this is where we're heading in terms of trying to understand the full intersectionality among self-identified race, socioeconomic status, sex, gender, age. So to wrap up, we see that brain health is really an important part of our overall health and well-being, and that even very subtle differences or decreases in our brain health as captured by MRI or cognitive test performance bode poorly for later clinical brain health outcomes. There are many cardiovascular and metabolic risk factors that are associated with lesser brain health across the lifespan, ranging from children to elderly. A lot of these risk factors that we know impact brain and cognition aggregate among persons of lower socioeconomic status. Therefore, perhaps it's not surprised that on average, people of lower SES are showing some signs of lesser brain health in our investigation so far. However, the associations of race and SES to brain health are complicated thus far. And just to remind you, as the examples, in our lower SES African Americans, there seems to be a particular vulnerability to the impact of poor diet quality and diabetes on cognitive functioning. Whereas our African Americans of higher SES are showing lesser brain health in other MRI-related dimensions, which we have not fully explained yet. But our higher SES whites are showing vulnerability to carotid atherosclerosis. So we are working on further understanding these complex patterns of association. So I'll put a plug in that we really need continued interdisciplinary collaboration to enhance our understanding of these very complex associations, and that ultimately this will hopefully lead to prevention and intervention efforts with the multi-level factors that impinge upon our brains and that allow us to preserve brain and cognitive health and functioning across the lifespan. I have to give some thank yous. Thanks for the dough to a lot of different institutions, and now the Roger C. Lipitz and Family Foundation. Many, many thanks to my collaborators at the National Institute on Aging's Intramural Research Program, especially my partners in crime on scandals and other handle stuff, Michelle and Alan. My many collaborators over the years from the University of Maryland School of Medicine and Baltimore VA, but particularly Les and Steve. These are the folks who've tolerated me the longest. Many wonderful collaborators at UMBC, University of Delaware, Oklahoma Health Sciences, Rochester, Penn, George Mason. And my lab, yay! So we have a host of wonderful graduate students in our lab working on these projects right now. We have a whole bunch of recent grads. When I say recent, I mean recent enough to have been here when we were starting the Handle study. So even if you don't feel like a recent grad. And then lots and lots of old grads, some of whom are depicted here. Thank you for the slide swipage and the late night assistance from some of you. And thanks to all of you for coming. And darn, it's hot up here. I'd be happy to take questions. Yes?
So that informs of this 20-year differential, I think, Roland Park on the Collins market, but then I was wondering, would you know how much of that might be due to differences in homicides and infant mortality rates? Because
Yeah.
Can you repeat the question?
Yeah, the question was, regarding that striking difference in life expectancy between Roland Park and Holland's Market, to what degree is that attributable to things like homicides and infant mortality rates? Some but not all. And I unfortunately cannot give you precise estimates. My colleague, Dr. Evans, would probably be able to do that. Hi.
I have a question that's about brain volume.
Yes.
Because from reading Stephen J. Kuhn, The Mismeasure of Man, I always understood that measuring brain volume meant nothing. So you must be doing something different than those craniologists did in the 19th century when they filled white skulls and black skulls with seed and measured and then decided that, white craniums were bigger and therefore white people must be more intelligent. So what is brain volume really about?
Brain volume is one of multiple dimensions of brain structure and function that is correlated with our information processing abilities. So there is kind of a long-standing is bigger, better question. And it is the case that across multiple studies, larger volumes are associated concurrently and prospectively with greater cognitive functioning. The presumption is that this greater volume in part reflects more dendritic branching, and so more extensive brain networks. But it is important to point out that caveat that these are not perfectly correlated, and so brain volume is one of many, many things that one wants to be considering in terms of a brain health outcome. The other thing I will point out in terms of brain volumes, lesion volumes we know absolutely are critically important. They predict stroke and dementia. However, the brain gray matter volumes, or the total gray matter and white matter volumes, are indeed predicting individuals who are prone to go on to develop dementia. They also predict mortality. And so our position is, just like very small increments in blood pressure, like even 2 millimeters of mercury increase in blood pressure at a population level translates into public health significance. Even these small variations in brain sizes and lesion volumes are in part capturing something that predict poor health outcomes. Yes?
Did you get a chance to look at the influence of diet or any of the behaviors like smoking and other kinds of things on some of these relationships?
Yeah, the question was whether we've had a chance to look at diet and or other behaviors like smoking on these associations. We have looked at first some very global indices of diet quality. These folks do 2 24-hour recalls, very detailed food diaries, which in this study that I talked about a little bit, we were looking at the Healthy Eating Index. And even the Healthy Eating Index is predicting certain aspects of cognitive function. We have not even gotten to the level of analyzing different macro and micronutrients that we know are pertinent to brain health. I will tell you, if you're interested in the diet of people in Baltimore, it's crap. Seriously, my colleague Marie Kuzmarsky, who is at University of Delaware, who is our Ph.D. nutritionist on this study, has a wonderful slide set showing the different major food groups that are consumed by people living in Baltimore. And I can tell you those include a sandwich category, burgers, Subway, things like that, fried chicken, pizza, most common vegetable is French fry, most common drink is a sweet drink. The healthiest subgroup of diet that's seen is rice, pasta, and some fruit. No one's eating veggies. But it's really very striking and sobering because this is crossing all socioeconomic strata. And Marie has associated publications that can be accessed on the Handles website. Smoking thus far we haven't been able to do much with because we don't have smoking topography in the Handles data set. As a large epi study, certain areas were trimmed more than others, and so right now we just have very gross measures of current ever-never, which is not capturing enough variance for us to understand what variation in smoking, alcohol consumption, and illicit drug use is doing. But even at that very gross level, the negative health behaviors do seem to be negatively impacting brain and cognition. It's preliminary data. Yes?
Yes.
Hi.
I have a couple of cardiovascular questions.
Okay.
Was heart rate ever a predictor of anything? The other one was when you talked about low blood pressure in a young group being a risk factor. I thought that was curious. Could you speak about that?
You're asking me about some curious behavior? So, first question, was heart rate predictive of anything? We've never looked at resting heart rate. I think it would be very interesting to do so because resting heart rate in part reflects vagal tone and poor vagal tone. Main dimension of our parasympathetic nervous system is known to be predictive of a lot of bad outcomes related to cardiovascular health and inflammatory processes. So, so the question is, I don't know, but it would be very interesting to look at resting heart rate and also heart rate reactivity. We have focused on blood pressure. The other comment was that it's a little curious that both high and low blood pressure were associated with poor outcomes. This nonlinear relation of cardiovascular risk factors to poor brain and neurocognition crosses multiple risk factors, and this is part of why we're having such a hard time with some of our statistical modeling with things like structural equation modeling, is that the relations are not linear. So across multiple samples now, we've seen that both high and low blood pressure, both high and low glucose, both high and low body mass index, and both high and low cholesterol are predicted of poor brain and or neurocognitive outcomes. Yes, yes.
I'm just wondering if there's any work on how people use their brains as affecting their brain health. So, for example, ways in which we use our mental capacities, like spiritual practices, or video games, or reading, or anything like that that impacts brain health.
Yeah, it's a great question. It's essentially is the use it or lose it. Is there any truth to the use it or lose it? It's not an area of work that I'm very familiar with the most cutting edge findings in that area. I believe there is some evidence for activity, brain activity, social activity, physical activity. It's hard to tease apart all of those, particularly in older people with respect to good outcomes. There's a very large national study experience core that tries to engage older retired people into volunteering in school systems to help tutor children, and are showing very good outcomes, presumably as a function of the increased brain, social, and or physical activity. We don't know how much to attribute to what. I am not aware of literature suggesting that video games are related to better outcomes, but it's kind of hard because that's also related to sedentary behavior, which we know is related to poor health outcomes. So it's seemingly an easy question, but I don't know that I have an easy answer. Yeah, It's a complicated question for a number of reasons. First, we've only been able to adjust for medication use right now because having someone tell you, I take Lipitor, doesn't mean anything because you don't know how adherent they are and whether they can afford the medication. So, there's a lot of kind of cross-sectional work looking at potential relations of different medications to cognitive outcomes, which are hard to interpret for a variety of reasons. What I can tell you is that there are a lot of randomized clinical trials out there as well that give people different meds and keep them on the meds. We've done antihypertensive medication clinical trials. My colleague Matt Muldoon from Pittsburgh has done multiple trials with various anti-lipidemic agents. The findings are decidedly mixed. So, with any clinical trial, it's really hard because it presents you average findings. On average, people didn't change or there wasn't any change, but that doesn't tell you about any subset who might be particularly vulnerable to medication effects for any reason. We don't have a good handle on vulnerability factors. Yes?
Hi. I'm wondering about, since you're studying brain and brain health, how did you screen in the Handle study for mental health, particularly given that not having received treatment for mental health or mental illness is no indication of its absence?
Right.
And then secondly, can you refer me to, or do you know of any really great work around the relations between mental illness and lower socioeconomic status? When I was in graduate school, I remember hearing sort of offhandedly from one of my profs that schizophrenia and poverty, for example, are very highly correlated, and people are beginning to wonder if it's not so much that schizophrenia leads people into poverty, but that perhaps poverty itself is influencing people's brain health.
Yeah. There are multiple questions that you've asked in literatures that I am not completely on top of. What I will tell you is that first, with respect to the Handle study, the only screening that was done is, has a doctor ever told you you had X, 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 and X? Okay, so a lot of people have never seen a doctor. The Handle's doctor is their doctor. So we assume that psychopathology is way underdiagnosed in this study. There has been a lot of talk. Actually, we were going to do a sub-study with a psychiatric interview, but could not kind of sort out what happens if we bring people here to campus and find someone who is suicidal or who really needs help, and where are we going to send them? And anyway, it hasn't happened yet, but there's a lot of interest in better documenting psychiatric illness in the Handle's participants. And there are screeners that are given for depression, PTSD, generalized anxiety, OCD, social phobia, psychotic symptomatology, things that suggest clinical cut scores, but we're not really sure whether they have equal validity in people of different socioeconomic or racial ethnic groups. The other question being, I believe, to what degree might mental illness drive people down to lower socioeconomic strata, and we assume that some of that is indeed going to be the case. So there's bidirectional activity going on there. I think I missed one part of the question.
Do you know of anybody doing good work in that area related to mental illness and poverty and lower socioeconomic status?
No, I would ask Jason Schiffman in our Department of Psychology. Yeah.
Is it an accurate generalization from the later material you were presenting that the traditional cardiovascular risks for neurocognitive performance are stronger predictors in your white urban sample than your African American urban subsample? Is that an accurate generalization?
No, it's that so far different types of risk factors seem to be more pertinent to different race by SES subgroups. And we're still really parsing out what is a predictor for what subgroups of people, and we haven't even started factoring in any detail men versus women, for example, and issues of nonlinearity, which we strongly suspect are operative in this sample, as we have people who are poorly nourished and who are taking drugs like cocaine and heroin, and so are going to have, say, better body composition profiles, better lipid profiles, for reasons that may not be so great. Yes?
For the socioeconomic status, did you look, for your participants, were they lifelong, lived in poverty, or did you also factor in maybe when they grew up they weren't in poverty, but now that they're adults, that they're in poverty? How that sort of works?
Yeah. Yeah, that's an excellent question. We don't have a detailed lifelong history of socioeconomic status. However, my colleague here, wave, Dr. Danielle Beatty-Moody, is looking at early life social disadvantage in our handles participants and how that is impacting brain health outcomes, and finding some interesting effects as a function of whether you are low SES in early life and change to high, or change from high to low, versus whether you stay stable. Yeah. Okay. Thanks. Would you like this?
No, I think I would.
You can be tied to the podium.
No,
now.
I think I won't. Let's thank Shari Waldstein again for this terrific, stimulating talk. I would also like to thank the Drescher Center and the Social Sciences Forum, and note that this has been the last talk in each of those series for this year. Am I right about that? And that you should look forward to listings of next year's information about next year's talks coming soon. I am also delighted to announce that the 2016-2017 Lipitz Professor in the Arts, Humanities, and Social Sciences will be Professor Anne Sarah Rubin of the Department of History. A brief introduction now, a much fuller introduction 1 year from now. Dr. Rubin joined UMBC in 2000. She's a historian of the American Civil War. In her 16 years here, she has published 2 books, winning major prizes for them. And to accompany her second book, which is called Through the Heart of Dixie, Sherman's March and American Memory, she developed an interactive website collaborating with Kelly Bell of our Department of Visual Arts and with our Imaging Research Center. Indeed, Dr. Rubin has been at the forefront of the field of digital history ever since the early 1990s, when she was a graduate student working on one of the pioneering projects in that area. Within the field of Civil War history, she has also been a leader within the field. She has served most recently as the president of the Society of Civil War Historians. I would note the first woman to hold that office. Dr. Rubin, yes? Dr. Rubin is also an accomplished teacher and mentor here at UMBC. Her courses include everything from large surveys to recently a small class that she co-designed with a computer science professor about historical gaming. She has advised dozens of students in the Historical Studies MA program, directing more than 20 theses and serving on more than 25 other master's committees in just the last dozen years. And in recognition of that work, she received the 2011 Donald Creighton Outstanding Faculty Award from UMBC's Graduate Student Association. Like Shari Waldstein, Anne Rubin exemplifies the combination of scholarship, teaching and mentoring, and leadership that is the hallmark of UMBC's and our college's most outstanding faculty. So congratulations to Anne on this award. We all look forward to your Lipitz lecture a year from now. And now I hope you will all join me in enjoying the food and the good company here. Thanks very much for coming.